The role of meiosis, then, is to generate haploid cells for sexual reproduction. Meiosis halves the chromosome number, but it doesn't just place any random half the chromosomes into each daughter cell. Normally, the cell that's going to undergo meiosis is a diploid cell, so it has two sets of chromosomes, and it will divide up those chromosomes by separating the homologous pairs in order to give one homologous chromosome from each homologous pair to each daughter cell. These haploid cells that result from meiosis are or will give rise to the gametes, or the sex cells. In animals, we're looking at sperm and egg cells. During sexual reproduction, a sex cell or gamete from each of two different individuals in the same species will unite to form a new diploid individual. They unite, of course, during fertilization. Where fertilization is between an egg and a sperm, half of the chromosomes will be of maternal origin. They would have been the chromosomes that came in the egg from the mother, and half of the chromosomes are of paternal origin. They would have been the chromosomes that came from the sperm cell or the father. And often in textbooks, you'll see the paternal chromosomes are colored blue and the maternal chromosomes are colored red. This movement between the haploid and the diploid state is part of the life cycle of most eukaryotic organisms. Humans and most animals spend most of their life cycle in the diploid state. So here we see the life cycle of a dog, and it shows you the movement between the haploid and the diploid state during the life cycle. In a regular adult dog, almost all of the cells are diploid. The only haploid cells are those that are produced for sexual reproduction. So we have a diploid organism. At some point, the germline cells will undergo meiosis to generate either egg or sperm. The egg and sperm are haploid cells, of course, and when they unite at fertilization, they generate the zygote, which is once again a diploid cell. And so the entire lifetime of the organism is spent in the diploid state. The haploid state only exists for sex cells. But lots of other organisms spend much more of the life cycle in the haploid state. Many yeast and other fungi, for example, exist as haploid organisms, and they move to the diploid stages only for sexual reproduction, which is often stimulated by tough times or moments of stress for the organism. Now, why might we see this move to sexual reproduction in times of stress? If the point of meiosis is to generate genetic diversity, then tough times is probably a really good time to do that, because if there's more diversity in a population, there's a greater chance that some individuals in that population will have mixes of alleles that render them better able to survive and reproduce in that stressful environment. This helps ensure the survival of the species and drives evolution. So how exactly do we generate haploid cells from diploid cells, and how do we generate genetic diversity? Meiosis is not going to generate or create new genes, of course, but it will create different mixes of maternal and paternal alleles. Looking at this process in overview, it involves two divisions. Prior to the beginning of meiosis, DNA replication takes place in the preceding S phase. So we enter meiosis with each chromosome consisting of two sister chromatids. In the first division, we have what is known as the reduction division. That is where homologous chromosomes are separated into daughter cells, resulting in the daughter cells being haploid, although the parental cell was diploid. This division is known as meiosis I, and again, the hallmark of meiosis I is the separation of the homologs. The second division is more like a mitosis, where the sister chromatids will be separated. This results in haploid daughter cells, in which each chromosome exists as a single chromatid. So the second division goes from two sister chromatids in a haploid cell to single sister chromatids in a haploid cell. This second division is known as meiosis II, and the hallmark of meiosis II is the separation of the sister chromatids. It's important to note that between meiosis I and meiosis II, the DNA does not replicate. So DNA replication, or S phase, occurs prior to the beginning of meiosis, and it does not occur between meiosis I and meiosis II. You should be able to look at this cell right now and tell me what is the value of N in this cell. Let's take a minute now and watch this animation that walks you through the stages of meiosis.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase I, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis II, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
I hope that overview is helpful. Let's just walk through that a little bit more closely now. Your textbook also does a really good job of describing meiosis, and I encourage you to read it as well. While the general stages of these 2 divisions in meiosis are similar to mitosis in that they also involve a prophase, a prometaphase, etc., what happens in terms of chromosomes themselves is different. As already mentioned, each chromosome replicates prior to meiosis to generate chromosomes, each of which consist of 2 sister chromatids. Given that this is a diploid cell which originated from fertilization, it has both maternal and paternal homologous chromosomes, and they're shown here. Prophase I of meiosis is where one of the major points of generating genetic diversity happen. And I mentioned earlier that meiosis creates different mixes of maternal and paternal alleles. This happens in prophase I through a process which is known as crossing over or recombination. In prophase 1, the homologous chromosomes find each other in the nucleus and they pair up. They align together along their length gene by gene. This process is known as synapsis, and it's facilitated by special proteins. After synapsis occurs, additional proteins come along and make breaks in the DNA in random positions. The repairing of these breaks is what leads to an exchange of DNA between the sister chromatids in the homologs, so that the end result will be sister chromatids, which are actually mixes of maternal and paternal DNA. Keep in mind that each chromosome is still existing as two sister chromatids. You should be able to look at this cell and tell me what is the value of N. What is the haploid number in this cell? The actual crossing over event is not well diagrammed in this image here, so let's look at a different image. Here we have two homologous chromosomes. They have aligned, and normally you're seeing only one sister chromatid aligned with the neighboring sister chromatid. In reality, these would be facing each other, so you would have all four sister chromatids interacting with each other. During crossing over, the result is that parts of what used to be on the paternal sister chromatid is moved to the maternal sister chromatid and the opposite. The outcome now, if you'll look here, is that you have 1, 2, 3, 4 sister chromatids, none of which are identical. You started off with 2 replicated homologs, and the 2 sister chromatids for each homolog were identical because they were generated through DNA replication. After crossing over, you have sister chromatids that have a mix of maternal and paternal DNA, and this means that all 4 chromatids will have different mixes of alleles and thus are genetically different from each other. This drawing has shown crossing over between only 2 of the sister chromatids, but in fact, crossing over will occur in all sister chromatids. This is just an image of actual homologous chromosomes paired during prophase, and you can see the points where crossing over has occurred. At the end of prophase 1, the nuclear membrane will break down and the cell enters prometaphase. In prometaphase, the chromosomes make attachments to microtubules to form kinetochore fibers. In terms of orientation, however, if you remember when we were talking about mitosis, each sister chromatid would become oriented to opposite poles. In this case, in meiosis I, each sister chromatid of a single homolog becomes oriented to the same pole, and the 2 homologous chromosomes remain connected or paired together. As a result, at metaphase, the homologous pairs come to lie at the metaphase plate. One of each of the homologs will be facing or oriented towards the opposite pole. During anaphase, sister chromatids remain together, and it's the homologous chromosomes which are actually separated, with one homologous chromosome going to each of the 2 poles. This splits the number of chromosomes in half for the daughter cells. So this cell here, you can see, has 6 chromosomes, and after anaphase, the 2 daughter cells will each have 3 chromosomes. Each of these daughter cells will be genetically different because they contain different mixes of the maternal and paternal DNA. You can tell this in this image by looking at the different colors on the chromosomes. The end of meiosis I then results in haploid cells with a haploid number of chromosomes, but each one of those chromosomes still consists of 2 sister chromatids. Meiosis II then is the division where the sister chromatids are separated. And meiosis II is most similar to mitosis for that reason. During meiosis II then, in prophase, the chromosomes will condense. In prometaphase, the sister chromatids will attach to microtubules at their kinetochores and become oriented to opposite poles. In anaphase II, the sister chromatids will be separated and will move to opposite poles, and this will generate daughter cells that are again haploid, but now have a single copy or a single chromatid for each one of the chromosomes. The overall result of meiosis will be 4 daughter cells, each of which are haploid and each of which is genetically different from each other. If you're finding this confusing, it's a useful thing to just draw out these stages. So give yourself a diploid cell with just 2 chromosomes if you want. Color them different colors, maternal and paternal, or give them different alleles for their genes on the chromosomes. Duplicate those chromosomes and walk them through meiosis. Sounds boring, but it really helps you cement your understanding. And let's end by watching meiosis in a living cell.
Watch how the chromosomes move during anaphase 1 of meiosis. This time-lapse video uses differential interference contrast microscopy to image part of meiosis 1 in a Drosophila spermatocyte. Notice how the chromosomes, which were lined up at the equator in metaphase 1, are pulled to the opposite poles in the cell of anaphase 1. Credit: Conley L. Ryder, Wadsworth Center, New York.
So if we back this up so you can actually see the moment of anaphase in case you're having trouble, let's just move this up to anaphase. So right here is where you can see the chromosomes are at metaphase, and these are homologous pairs, right? These are not just single chromosomes with 2 sister chromatids.
Differential interference contrast microscopy to image part of meiosis 1.
There, this is the moment of anaphase. You can see here's one group of homologs and the other group of homologs there, moving towards the poles. This is time-lapse, so it doesn't happen this quickly.